Hi, my name is Jane Picarelli, and today we're going to be talking about glycolysis, gluconeogenesis, and aldolase. I hope you enjoy this presentation. So we have a lot to go over today, so I'm going to give you a light outline on what we'll be talking about. The heaviest emphasis on this presentation is going to be glycolysis, but we will also be covering a few more things. So after an introduction, I'm going to talk a little bit about key terms and concept involved in the processes we'll be covering so that as we're talking about it, you could follow along a little easier. I'm going to include an animation before we go through the steps of the process, just so that when we do go through the steps, you can kind of put it in context of what's going on. We'll then talk about the step-by-step reactions of glycolysis, and then we'll go into the basics of gluconeogenesis, and we will follow that with the significance of aldolase and what its function is. All right, let's go. So I don't know about you, but in my younger days, I could eat a whole pizza by myself. I could also eat about 8 tacos by myself. And as you know, what goes in as a whole pizza or 8 tacos does not come out as a whole pizza or 8 tacos. So just as in it does not come out the exact way we ate it, the food that we eat does not directly turn into energy either. It has to go through quite a bit of processes, and one of those processes is called glycolysis. Glycolysis is composed of 2 words, the first meaning glyco, which means sweet and references sugar. The second means splitting, and that term is lysis. So this process will be the splitting of a sugar molecule, and we'll get to that in a bit. So as a person who never got to take chemistry in high school or in junior college, and having my first experience with it in my undergraduate studies, and then moving on to doing it in my master's program, there was a lot of topics I was confused about. So I want to clarify a couple basic terms and a couple basic processes that are going to be used in this video to help those who maybe aren't as familiar with some of the processes involved. So this is adenosine triphosphate, also known as ATP, and this is a molecule that carries potential energy. So it consists of an adenine, which is this right here, a ribose sugar, which is this right here, and 3 phosphoryl groups. And a phosphoryl group just means a molecule that has phosphorus and oxygen. So if you're looking at that, you can see the phosphorus in the center and the oxygen on top. So ATP gets its name from these 3 phosphoryl groups. So the group closest to that ribose sugar right here, that's going to be the alpha group. The second one is the beta group, and this last one is known as the gamma group. So these bonds are actually high energy bonds, and they're called phosphoanhydrate bonds. And through hydrolysis reactions, they can actually be broken. And when they are broken, which will look like this, when they are broken, this molecule, because that last phosphoryl group broke off, this molecule actually changes and becomes adenosine diphosphate. And that di is for the 2 phosphoryl groups that remain. And what happens is that phosphoryl group that broke off, it actually gets transferred to another substance, giving that other substance its potential energy. And in that way, it's like an energy wallet molecule, the ATP, I mean. So the leftover adenosine with the 2 phosphoryl groups is called ADP, or adenosine diphosphate. ATP is actually an unstable molecule, and it's able to spontaneously break apart. So one thing you need to know about the ATP, again, that's when it has the 3 phosphoryl groups, you need to know that it must be coupled to reactions that require energy at the same time, because as it's losing its 3rd phosphoryl group, for that phosphoryl group to be of any use to anything whatsoever, it needs to be coupled to the reaction or the substrate that needs the energy for it to occur. So in the center of this image, you see in black the word kinase. A kinase is an enzyme that enables the transfer of one of those phosphoryl groups from one substance to another. So in this presentation, you will see that occurring as a phosphoryl group gets transferred from ATP to its receiving molecule. So the process that we just saw on this previous slide, it was carried out by a kinase, as mentioned, and the process itself is called phosphorylation. So this is where a phosphate group gets transferred from one molecule to another by a kinase. That's all that means. And this actually usually occurs in some processes that involve some sort of cellular regulation or a potential energy transfer. So hydrolysis comes from two root words, hydro, which is referencing water, and lysis, which, as we talked about earlier, means splitting something. And it's just where you have some sort of molecule or reactant, and it's in the vicinity of a water molecule. And the water molecule is going to split into a hydrogen and a hydroxide, which is an oxygen and a hydrogen. And each of the parts that the water splits with is going to bond to part of that original molecule. And so in that process, it also splits that original reactant. Certain arrangements of molecules get designated as part of a group. For example, earlier I mentioned that the phosphoryl group is a group of molecules that contain phosphorus and oxygen. Well, a carbonyl group is a group that has a carbon double bonded to an oxygen, as you can see in the picture to the far left. The X's represent any other molecules bound to it, as those are not significant in the definition. Two specific molecules that have the carbonyl group in it are ketoses and aldoses. Aldoses have one aldehyde group per molecule, whereas ketoses have one ketone group per molecule. In the bottom right-hand corner of the slide, I added an image of the structure of an aldehyde and a ketone for reference. They all contain carbonyl groups. So just to show you with a little visual, glycolysis is a 10-step process that occurs in two main phases. One of the phases is going to use energy, and the next phase is going to produce energy. And there's several names for each of those phases. The first phase where it uses the energy, some people will call it the investment phase, and some people will call it the preparatory stage. And the second stage is called the payoff stage. So whatever you want to call it, it starts with a molecule of glucose and ends with the generation of two pyruvic acid molecules, two NADH molecules, which are those electron-carrying molecules, and 4 ATP. But it's worth noting that because 2 ATP get used up in the investment phase, there's only a net of 2 ATP created. Now the pyruvic acid will continue on to a reaction that occurs after glycolysis. So that's why that's important. And again, ATP is an energy carrying molecule. So that's why that's important. So this is a very, very basic animation. In each of the molecules and how they're represented, they're a lot more complicated in real life than that. We will go through the actual steps, but just so you have a super, super broad overview of what's going on, this way it'll kind of help you put it in context when we do actually go through in depth the reactions. So, using my laser, here, these 6 carbons are representative of a molecule of glucose. These are representative of electrons. These are representative of those ATP molecules that we talked about, and this is the 3 phosphoryl groups. The little boom is just showing energy. NADs are an electron carrier, and then in the animation you will see how they turn into NADH when they receive electrons. Again, this is going to be extremely basic, just to help you get the concept down before we move along. So you have your glucose, and then you have transfer of the phosphoryl group of ATP, and it leaves it with ADP, which gives it energy, not from the breaking of the bond, but the potential energy in the phosphate. So after that, with that energy, it allows this to be split, but these two leftover molecules, the 3 carbons on each side, were not at pyruvate yet. So either way though, the molecule is no longer glucose. Next, the electrons change the NAD plus into NADH, which is an electron carrier. And then finally, ATP is regenerated with a phosphoryl group transfer. And after that, you have 2 molecules of pyruvate. So on the next few slides, we're going to cover the investment phase, also known as the preparatory phase, because this next couple of steps actually uses energy. So we'll go a little bit more detail than the overview covered. So in step 1, glucose, which is a 6-carbon sugar, is phosphorylated. So the present ATP molecule is the molecule that donates its phosphoryl group, and in this, 1 molecule of ATP gets consumed. So the reaction in this step, which again is where the glucose gets the phosphoryl group, is made possible by the kinase hexokinase. So hexokinase binds to glucose, and it undergoes a conformational change which lessens the distance between ATP and glucose, and that protects water molecules from breaking ATP's phosphoanhydride bonds via hydrolysis prematurely. So you wind up with, at the end of step number 1, glucose 6-phosphate. So now in step 2, an enzyme called phosphohexosisomerase serves as a catalyst to isomerize the glucose 6-phosphate from the last step into fructose 6-phosphate. So in step 3, phosphofructokinase 1 is responsible for the catalysis of a reaction which results in a phosphoryl group being transferred from ATP to the product of the previous step, which was the fructose 6-phosphate, resulting in fructose 1,6-biphosphate. PFK1, which is the phosphofructokinase 1, increases in response to a low supply of ATP, and the opposite is true for the converse situation. But again, to refresh, at the end of this step, you have fructose 1/6-biphosphate. In step 4, aldolase, which is short for fructose 1,6-biphosphate aldolase, initiates the lysis, that splitting portion of glycolysis, which is a reaction that results in fructose 1-biphosphate being cleaved into 2 3-carbon molecules. The aldolase reaction that causes this will further be discussed when we get to the section about aldolase later in the presentation. So once cleaved, the remaining molecules are named glyceraldehyde 3-phosphate, which is an aldose, and dihydroxyacetone phosphate, which is a ketose. So this is going to be the last step in the investment or preparatory phase of glycolysis, and 2 ATP molecules have already been used up so far in this reaction. So recall that the 2 molecules that were generated in the previous step were different from one another. This may be confusing since we know that the end result of glycolysis is going to be 2 of the same molecules, those pyruvates, but here's how this is possible. So the glyceride 3 phosphate will eventually be broken down later in glycolysis, while the dihydroxyacetone interconverts between itself and glyceraldehyde 3 phosphate. So this interconversion reaction is catalyzed by the enzyme triose phosphate isomerase. And at this point, due to the fact that the dihydroxyacetone phosphate is equally in a state of being glyceraldehyde 3 phosphate, it can be said that at the end of the preparatory phase, the original glucose molecule has been yielded into 2 molecules of glyceraldehyde 3 phosphate. So now we enter the payoff phase of glycolysis. So in the previous phase, remember that we used up some ATP. Now we're actually going to create some. Glyceraldehyde 3 phosphate in this step becomes oxidized to 1,3-biphosphoglycerate with the help of the enzyme glyceraldehyde 3 phosphate dehydrogenase. This is part 1 of 2 reactions in glycolysis that conserve energy and contribute to forming the adenosine triphosphate. The specific part of glyceraldehyde 3 phosphate that gets oxidized is its aldehyde group. NAD+ in this step becomes NADH. These molecules are important because it stores the electrons that are lost in oxidation. They're electron carriers. Next, an enzyme called phosphoglycerate kinase transfers a phosphoryl group from 1,3-biphosphoglycerate to ADP. Now, if you remember from an earlier slide, I discussed how when ADP, because that di stands for 2, when it receives another phosphoryl group, it becomes adenosine triphosphate. Recall tri means 3. In other words, in this step, ATP is formed. In addition, 3-phosphoglycerate also remains. Note, this was part 2 of the energy conserving reactions as I mentioned in the last step. Step 6 and step 7 are energy coupled reactions because part of the 1,3-biphosphoglycerate formed in step 6, specifically its acyl phosphate group, is what's transferred to the ADP in this step. The 3-phosphoroglycerate from step number 7 in this step gets converted into 2-phosphoroglycerate with the aid of enzyme phosphoglycerate mutase. This enzyme enables the phosphoryl group being transferred from glycerate C2 and C3. Now in step number 9, Aldolase reversibly removes a molecule of water from the 2-phosphoglycerate to form PEP, which is phosphoenolpyruvate, and this is a dehydration reaction. So in this final step of glycolysis, a phosphoryl group is transferred from the phosphophenolpyruvate in the last step to ADP. The enzyme that catalyzes this is pyruvate kinase. The products are ATP and pyruvate. So you may be wondering why glycolysis is so important. Well, the pyruvate that was generated by the end of glycolysis goes on to the next steps in cellular respiration when oxygen is present. It has some of that potential energy from the glucose molecule. And later on, when oxidative reactions in the citric acid cycle take place, that potential energy can be used. And in addition, the ATPs that were generated, those are potential energy reservoirs. And the NADH, those are going to be your electron carriers. So most of the products from glycolysis go on to be used for later reactions. Gluconeogenesis takes place mostly in the liver and occurs when existing glycogen stores are low and when carbohydrates are not being consumed. This might be during times of starvation or a low-carb diet. I'm not going to dive as deep into these steps as I did with glycolysis because many of the steps are the same but in reverse as those in glycolysis, although there are definitely differences. The most important thing to know about gluconeogenesis is that it occurs at times when carbohydrates are scarce and it exists as an alternative means for the body to avoid becoming deficient in glucose. Lactate, glycerol, and amino acids, which can form pyruvate, are the necessary precursors for initiating gluconeogenesis. One of the reactions in gluconeogenesis actually happens in the cell's mitochondria, but then, like glycolysis, the rest occurs in the cytosol. There are three main steps of gluconeogenesis. They include pyruvate being converted into phosphoenolpyruvate, which ends with fructose 1,6-biphosphate for step 1. Step 2, fructose 1,6-biphosphate is converted into fructose 6-phosphate and ends in glucose 6-phosphate. And step 3 is what allows for free glucose to enter the blood, and that's when glucose 6-phosphate is converted into glucose. Pyruvate is made from amino acids such as alanine and lactate. It is catalyzed by pyruvate carboxylase to become oxaloyl acetate. It is worth mentioning that because this step occurs in the mitochondria, which the oxaloyl acetate cannot cross. It must be converted into malate or aspartate temporarily to cross the membrane into the cytosol. Once it is reformed in the cytosol, the oxaloyl acetate is converted to phosphoryl pyruvate when the phosphoryl pyruvate carboxyl kinase removes the carbolic acid from it. Next, through a series of reversed reactions that occur in glycolysis, a fructose 1-biphosphate is created. In the second main step of gluconeogenesis, the enzyme fructose 1,6-biphosphatase catalyzes a reaction that converts fructose 1,6-biphosphate into fructose 6-phosphate. The fructose 6-phosphate is then converted by the same isomerase from glycolysis into glucose 6-phosphate. An enzyme called glucose 6-phosphatase catalyzes a reaction that results in the glucose 6-phosphate releasing an inorganic phosphate, allowing the now free glucose to enter the blood. In the presentation outline, I referenced the fact that I would be discussing aldolase. So if you're wondering why, I will be explaining its significance shortly. Aldolase is actually short for fructose 1,6-biphosphate aldolase. Yeah, that's a mouthful, and that's why we just call it aldolase. But it's an enzyme that plays a critical role in both gluconeogenesis and glycolysis. Remember in glycolysis, there's a step where the fructose 1,6-biphosphate is split into a glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. Now, do you remember the long name of aldolase? That's right, it's fructose 1,6-biphosphate aldolase. And it's named as it is because it is involved in the reaction that leads to the cleavage of the fructose 1,6-biphosphate in step number 4 of glycolysis. The reaction itself is an aldolase reaction, which is the reverse of an aldol condensation reaction. And if you look at the image, you can actually see that here you have the fructose 1,6-biphosphate, and then this is where the enzyme aldolase, again short for fructose 1,6-biphosphate aldolase, comes into play, and it catalyzes this reaction, this aldolase reaction, and it yields a molecule of glyceraldehyde 3-phosphate, and a molecule of dihydroxyacetone phosphate. So as I alluded to earlier, aldolase is also involved in gluconeogenesis. You don't have to go back and rewind this video. This is the same picture I posted in the last slide. The aldolase reaction is reversible. The body's needs and availability of certain molecules determines the direction that this reaction proceeds. In gluconeogenesis, glyceraldehyde 3-phosphate and dihydroxyacetone already exist, and the aldolase catalyzes the joining of the molecules by an aldol condensation reaction to make a 6-carbon molecule, that fructose 1,6-biphosphate. Aldolase is important in this step because the 6-carbon molecule will then undergo more reactions and become glucose. The 3 isoforms of aldolase are aldolase A, aldolase B, and aldolase C. They are encoded by 3 different genes. The different isoforms are expressed in different parts of the body and are associated with some overlapping but some independent processes and functions. Deficiencies in these isoforms leads to pathology. This is a little chart that I compiled to highlight some of the key differences between the isoforms. So, for instance, with aldolase A, it's located mostly in the muscle and other tissue. It's involved in fetal glycolysis, and a deficiency in it can cause mental retardation, abnormal stature, and hemolytic anemia. Aldolase B is found mostly in the liver, kidney, and small intestine. It's regulated by fructose exposure, and a deficiency in it can lead to hypoglycemia and a fructose intolerance, which can be observed by if an individual with a deficiency in this aldolase B eats any fruit sugar, fructose, they will start vomiting. And then lastly, aldolase C. That one is mostly found in the brain, and it has a special ability to where it can also convert fructose 1-phosphate into DHAP and glyceraldehyde. Aldolase B expression in the liver can be regulated by transcription factors HNF1, HNF3, CEPD, and DBP. That's a tongue twister. A transcription factor is a protein that affects the rate or process of making messenger RNA from DNA. That process is called transcription. When someone speaks of turning genes on and off, they're usually referencing the work of transcription factors. Expression of aldolase B in the liver is related to several transcription factors. HNF1 and HNF3 are transcription activators. CEPB is a transcription regulator and weak transactivator of the aldolase B promoter. HNF1 strongly transactivates aldolase B promoters. HNF3 acts in strong opposition to the activation of the aldolase B promoter. In this presentation, we have discussed some key terms and topics that provided background information for understanding the metabolic processes. We then dove into the 10 steps of glycolysis and the 3 main steps of gluconeogenesis. From there, we covered the role that aldolase plays in both processes and reviewed its different isoforms and their expressions. We then talked about the transcription factors involved in aldolase B expression.